Bighorn Sheep to South Dakota for study. Dr. Wolf.
This is a PowerPoint.
PowerPoint. We'll move down here.
Sorry.
It's timeout.
Are you ready? Are you ready? Yeah? Okay. For the record, Dr. Perry Wolf, veterinarian, Nevada Department of Wildlife. Earlier this spring, about February, I was approached by the Eastern Region biologist, Matt Jeffers, about whether there might be interest in the research community about sending some of our snowstorm herd sheep for potential study in disease issues. So I put that out to a couple of the prominent researchers in bighorn sheep diseases and immediately got a response back from Dr. Tom Besser at Washington State University indicating that they would be interested in a number of sheep for a study that would explore the role of what they're calling super shedders in the persistence of disease in bighorn sheep herds. So as we've just been talking about, we know that a disease event is pretty devastating, but it's really the most devastating thing about this is the fact that lamb recruitment in subsequent years, and I think the longest one that Mike had on his list was 28 years with poor lamb recruitment, often at 0% survival. There's been some work from Hell's Canyon, which is on the Idaho-Washington-Oregon border and is probably one of the biggest well-studied metapopulations of bighorn sheep with disease in the U.S. And through Washington State University with some of their pen trials for domestic sheep, that when they've gone back after a die-off event or infecting animals with MOV and consistently tested individual animals over time, that they found that they break out consistently into three categories, and that an individual sheep will continue to test negative for MOV over and over again. There seems to be a group that will intermittently test positive and then test negative again and then maybe test positive. And then there's also a group of individuals that appear to be chronic shedders and continually test positive. And that might be, 4, 5, or 6, 7 samples that they've had on these sheep over a number of years, both in some of these domestic sheep and wild sheep. So there's the thought now that after a die-off event, that of the surviving adult sheep that are there, that potentially anywhere from maybe 10 to 20% of those adults remain as chronic shedders of MOV. So their research hypothesis for this study is that it's these chronic shedders that maintain the respiratory disease and cause it to persist in these bighorn populations, and that through contact with these animals is why the lambs are getting sick and dying. So it's these animals that are there. And the hypothesis is that if we remove chronic MOV shedders from the population, is there a potential that we can increase lamb survival in the herd? So they've devised a couple of experiments. This one is a captive experiment, South Dakota State University in Brookings, South Dakota. It's a commingling study to monitor health and survival of lambs born to ewes of different known shedding status of MOV. They've designed it for 3 replicate populations of 10 sheep total from infected populations, and then they would look at these population responses within these commingling studies over 3 years. They've started the study with a small group from the Hells Canyon population already this fall, and so they moved those animals in late winter. The basis of the experiment is in year 1 that we would have these ewes in a pen that were consistently negative for MOV. You would also have a group, and these would all be pregnant ewes, that are considered intermittent shedders, and the feeling is that those animals, once they're away from chronic shedders, will eventually go to become negative shedders, and that you'll have a group of chronic positive shedders. The thought is that in the negative group there will be lamb survival because there will be no transmission of MOV to lambs. The intermittent shedders, when they're not exposed to chronic shedders, will become negative and their lambs will survive. And then the lambs of the chronic shedders will die from pneumonia. In the second year experiment, they will be bred back to a ram from the same herd so that we're not potentially mixing in or introducing any new pathogens to those ewes from this population. So they'll take 2 of the positive chronic shedders and put them in with the negative ewes. They'll take 2 of the chronic shedders and put them in with the intermittent, hopefully negative ewes. And the prediction is that all lambs will then get pneumonia and die because they will have gotten infected from those chronic shedders. Then in year 3, they will separate them back again, put the negative ewes in their own pen, put the intermittent ewes in their own pen, and put the chronic shedders back in their own pen. And again, we expect to see the same results, that the 2 negatives will have live lambs and that all of the lambs in the chronic shedders will then die. So we are proposing that we take animals from the Snowstorm Mountain herd. This herd had a documented die-off in 2011. Since then, the biologist has indicated and recorded that lamb recruitment has been essentially 0. There's approximately 40 animals remaining in that population. 12 of those animals are collared and have known MOV status data, which is very critical to this because we can then we at least know who's negative and who's positive, and then they can sort those out. So these sheep would be moved from the snowstorms to Brookings, South Dakota. Our plan is to try to do this in November of 2014 after the breeding season so that we're already moving pregnant ewes. They're looking for 10 to 12 ewes and then a ram that can stay there so that he can breed back to the ewes every year for part of this. South Dakota State University and Washington State University have come up with money to pay for the initial capture of the animals, and then also will be responsible for transport of those animals to South Dakota. And then we feel strongly that we would like to redeploy those collars on another 12 sheep in the snowstorms, and our plan is that NDOW will pick up the tab for that. So that is all I have for that portion of the presentation. Do you have any questions?
Any commission questions?
Mr. Chairman, I've got a
Commissioner
question.
Lane?
I'm just trying to think how to phrase this question. Has there been any studies where the chronic shedders have actually been destroyed to see what happens with the population at that point? I mean, given the fact that we had a die-off, so has anything been done in that area?
To date, no. One of the things that prompted Iowa, or Idaho, and South Dakota and Washington State to kind of put this together was the fact that, as Mike alluded to, we've been chasing pathogens, we've been looking at that. I think we have a pretty good handle on what kills bighorn sheep right now. The big frustration out there that's been voiced over and over again by the animal managers and all of us is, okay, well, now we got a sick herd, now what? We sit there for 28 years and just watch this thing go down to zero. So they put this together, and in Hell's Canyon, they have, as I said, they've got a tremendous amount of data on that. So they may, in concert with these Penn studies, start looking at trying to identify chronic shedders, since they have many of them on paper in their own herds, and start eliminating those animals. We've had some preliminary discussions whether we could try something like that in some of our herds that have had multiple years of poor lamb recruitment. One of the general frustrations is if you go into this kind of test and cull type of protocol, which is very common around the domestic livestock, been used for years and years, but our problem is that we don't really have a bedside test for telling if an animal is positive with MOV or not. But it still may be worth trying in a small herd type situation to see if we could actually achieve the same thing while we're waiting for the results of these Penn studies, because you're looking at 3 years out to really confirm stuff in a controlled environment. Maybe we could also look at doing some of that in concert in the field, and they're definitely planning that in Hell's Canyon.
Any other commission questions? Commissioner Macbeth?
Perry, so within a population, there can be the 3 groups within a single population.
Yes.
So with respect to the ones who never have the disease, what is your belief as to those sheep? Is it because they just simply don't come in contact with the diseased sheep, or are they? Is there a thought that there might be some immunities or something there that are keeping them from getting the disease? And has there been any work in that area to explore that?
I think there may be that they're not 100% sure whether they never become infected or whether they become infected but are able to clear the disease. And I think if you look at disease dynamics across all diseases and all species, including humans, if we were all exposed to a disease, there'd be certain people that would die, certain people that may become chronically infected if the disease acts that way, some people that may temporarily have the disease but clear it and then go on and be fine. And so the thought is that these chronic shedders, and I don't know that there's not, to my knowledge, a good understanding of what causes some of these animals to remain negative persistently over time in the face of the fact that they have cohorts in the group that are chronic shedders. Because certainly when you look at Hell's Canyon is California bighorn sheep, and they tend to cluster a lot during the winter time on winter range, and I think there's certainly ample opportunity for ewes that are chronic shedders to come into contact with ewes that are negatively infected, and for some reason they clear that infection, and no, they do not understand why. And they have also had 2 ewes in hand, both fat, healthy butterballs. One, when they culture her, she's shedding MOV, and the other one is not. And just from looking at them from the outside, you can't tell which one is which is frustrating. And this is, I think that it's important for us to understand the mechanics of MOV. We don't understand how it fully behaves in bighorn sheep over time. The only result that we usually see on the ground is healthy lambs that are born, and at about 6 to 12 weeks, like clockwork, they start getting lethargic, they start getting sick, and boom, they die. And there's been a number of studies in Colorado with 2 of their herds that had been long-term poor lamb recruitment where 100% of the lambs died. They opened up all of them, and all of them had pneumonia. And the theory is that these 10% of these chronic shedders are exposing the lambs, through nose-to-nose contact, or one of these is the mom of one lamb, and then when they get in their nursery groups, they spread it around and end up dying. But why the other ones don't get it, no, we don't. There's not enough immune function studies of bighorn sheep to understand that.
Commissioner Drew.
Could you give me just a little background on the pen studies and how that's worked in terms of going from animals that are in a wild setting to a pen setting? And it sounds like it's been done. So, I mean, is there any effect in terms of going from those two kind of extreme environments, and what's been the experience on that?
They've been doing pen studies since the 80s up in Washington State University, and there's a number of other agencies that have captive sheep. And I think if you have spent any time at Hemingway Park in Boulder City, I think that, we call them wild, but I think a lot of times they tend to calm down quite easily, breed readily in captivity, and I personally don't believe that it's a huge stress on them. I think the biggest thing is, as long as they're looking at another sheep, then they're fine, and all their biological needs are met. And they do things, make sure they can, have some places to hide and that sort of stuff, and take precautions because they are wild animals, but they do remarkably well.
Anybody else?
I got
Commissioner Bliss.
just one question. What's the time frame from the time that you collect the samples and then you send it off to get your results back? And the reason I ask that is if we were to try it in one of our small populations, what would be the means of holding those captured sheep in a spot to where you would get those results, to where you could cull the ones that you needed to cull and some that you didn't? I was just curious on that time frame.
We usually, if you were probably going to push it, we take the samples in a broth, and then that broth has to incubate so they can actually get the DNA of the organism. And I believe that they incubate those for 72 hours and then run what's called a PCR test to isolate the DNA out of that. So, if you could get it to the lab in 24 hours, you're probably looking at 3 to 4 days if you could really push it. What they're working on now is trying to develop it so that you can just take the swab, and you don't have to put it actually in an enrichment broth, but you can take that swab and run the DNA tests on that. But at the moment, that technology is not available.
Anybody else? Do you have anything to add before we go to public comment?
I just think that this is a great opportunity for us to contribute sheep to hopefully, work slowly toward a solution to when these animals become infected, rather than just being stuck and watching a herd slowly decline. If there's anything, we may be able to develop a test that would let us understand at the moment that a sheep is positive for MOV, but until we understand if it is these super shedders that are causing the issue, then we're still in the dark about what happens after the die-off and why we continue to have these years and years of lamb recruitment. So I think that this will, if it doesn't immediately result in a practical test and cull solution, it will get us that much closer to understanding how the disease manifests on the landscape, and I think that's critical.
All right. Any public comment? Come on up.
Deborah Stowwater from Las Vegas, Nevada, Clark County. I just wanted to say I applaud the lady here. Perry? I applaud Perry for her consideration of this. I think it's a good way of us as on the Wildlife Commission and everything, supporting this commission to show the other states that we're willing to work with them. Hopefully they'll be willing to work with us in the future on other projects we need.
Thank you. Any other public comment? Come on up.
Craig Burnside, Douglas Cap. We unanimously voted to support this action, and the discussion that we had was just echoed by Dr. Wolf's last sentiments, that, it's frustrating to sit and watch this happen. And so if there's anything we can do to try to prevent it in the future, or at least curtail it in the future, we're willing to support.
Anybody else? Seeing none, I'll bring it back to the commission. Commissioner Drew.
Motion to approve the request for translocation of California bighorn sheep to South Dakota as presented.
Second.
We have a motion by Commissioner Drew, second by Commissioner McNinch. Any further discussion? Seeing none, I'll call for the vote. All those in favor signify by saying aye. 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 All those opposed? Okay, motion passed unanimous. We're going to move on to agenda item number 8, duck stamp request. Mike and Mark.